Okay, I invite the next speaker, Dr. Jyotsna, to talk to us about toll-like receptor 4 in acute myocardial infarction.
Good morning to all, and thanks to the organizers to giving this opportunity. The main problem with an acute MI is with all the gadgetries, including the primary PCI and the drug therapy, still the mortalities are high. So still we have to work on the subcellular level. What are the modifications is required in addition to the pharmaco-invasive therapy, which is already previously insisted. For several decades, it is known that the inflammatory response during the reperfusion is a major cause of the myocardial injury. Therapeutic options are limited by the lack of the understanding of intra and intercellular mechanisms between the inflammatory cells and the cardiac myocytes. Above all, there is no specific therapeutic target for inhibiting the inflammatory responses in which the cardiomyocytes are involved. The identification of the toll-like receptors, that is in short can be called as the TLRs, have rise to not only to the insight of the inflammatory response initiated by the cardiomyocytes themselves, but also provided a potential targets to reduce the myocardial injury. Mostly the TLRs are the proteins which are activated against the microcytes, antimicrobials, as a first inflammatory mechanisms. But subsequently it is demonstrated that the different types of the TLRs, which are especially present on the plasma membranes, that is type 1, 2, 4, 6, are the very important in the prevention of the myocardial injury. Like when the glycoprotein 2b3 receptors are detected, this was mainly for the pathogenesis to know the thrombogenesis. But subsequently, the glycoprotein 2b inhibitor receptor blockers has become a therapeutic modality of the treatment. Like that, we are anticipating that the probably toll-like receptors may be at the initial stages as a response to the acute MI, but subsequently the blockade of these receptors may be one of the therapeutic options in acute MI management. The primary aim of this study is to see the level of the toll-like receptors concentration in acute MI patients. We have taken from the mononuclear cell concentrates and also from the plasma. In subsequent slides, I will say that why we have to distinguish like directly calculating from the plasma versus mononuclear cell concentrates. And the second component is the TLR4 is a marker for the in-hospital events, that is the primary endpoints, which are the angina, re-MI, left ventricular failure or death. Of course, we are not included as a primary VT in these cases. Secondarily, we wanted to compare the already existing inflammatory marker, that is hsCRP, and another important marker of the acute MI for the prognosis is the cardiac enzymes. So we wanted to compare the TLR4 concentrations with the hsCRP and the cardiac enzymes. The inclusion criteria is an acute MI age group between 18 and 80 years and acute MI between 6 to 48 hours of duration. As we already know that within the 6 hours, which are the best ones, so we don't want to include them, with or without thrombolysis, with or without the primary PCI, with or without the cardiogenic shock. We included all the complications along with an acute MI because that will be one of the events which we require for the prevention in the subsequent hospital stay. That's the reason we included all the complications associated with an acute MI. Of course, exclusion criteria, the patients who have their significant underlying other systemic disorders like a chronic renal failure, hepatic or respiratory failure, or a malignancy which is limiting the life expectancy, and acute cerebrovascular accidents, which may be associated with an increase of these inflammatory markers. So we calculated both from the plasma as well as from the mononuclear cell concentrates, the toll receptors. These toll receptors are present on the surface of the plasma membrane, and they're mainly the previous studies had demonstrated isolation from the lymphocytes plasma membrane, that is from the mononuclear cell concentrates, and either directly the protein estimation or mRNA levels from the lymphocytes. The major problem there is it takes a longer time. The mononuclear cell concentrates by Ficolpec method, it takes about at least 6 to 12 hours. This is actually the blood sample collection is done, and we combine along with that with the Ficolpec and centrifuge it, and the supernatant fluid has to be taken off, and then the pellets are to be taken, which has then enriched with the mononuclear cell concentrates. And the lymphocytes are the component of these mononuclear cell concentrates. So this is the method which is previously done by the many of the investigators, and the TAL receptors, all the studies are demonstrated, either the acute MI, chronic stable angina, or the post-CABG, congestive cardiac failure, these TAL receptors as a prognostic markers are proved. But the major problem there is it's not the determinant, either the prognostication or for the therapeutic modality. But in our case, it is what we require is in the acute condition, how we are going to use it. So we have taken one group, the 20 patients, from whom we isolated the mononuclear cell concentrate and estimated the TAL receptors, and the other one is from the plasma. Once it is expressed on the cell surface, they are secreted into the plasma when the certain threshold is reached. So they will be detected from the plasma also. Especially this idea has come when from the plasma using from the already bacterial infection patients are tested, from the plasma high concentrations are detected. So we thought that it may be the true even in the acute MI also. Once the concentration is more, probably they will be secreted into the plasma. And instead of doing all the major procedure like a separation of the mononuclear cells, which takes a longer time, and then doing it, in the group B, we started using the plasma of the patients of the acute MI to detect the TAL receptors. So the number of the patients, this is an ongoing trial, and I am presenting it is a single center trial and investigator directed trial, and no funding agency for this. And the number of the patients we recruited till now are then about, I analyzed in about 40 patients in the cases and the 20 controls. We have taken the 20 controls because mainly to know about the circulating levels are the present in the normal patients, especially when they have the diabetes and hypertension. That is another important risk factors which are known to be associated with increased HSCRP. So we wanted to exclude those conditions. That's why we have analyzed in the 20 patients also, with the average age group of the 55 years, and male is to female ratio is the 4:1, and hypertension and diabetes is the present in about 52 and 25%. And Clelips class was 1.7 plus or minus 0.9, and these are the CPK levels, and TLR4s are about 0.07 to 0.04 and not detectable in the case of controls, and HSCRP was a 3.3 in the case of the cases, whereas the 0.08 plus or minus 7 in the case of controls. And as already mentioned, these are the locations of the MI, that is an anterior MI in 65 and then inferior in the 27, and extensive, that is involving the both anterior and the inferior in the 7.5%. And 14 patients undergone the primary PCI in these groups, and early PCI was done in 18 patients. So this is the group A and group B. Group A is where we calculate the TLR from the mononuclear cell concentrate, and the group B from where we calculated from the plasma. From the plasma, actually the concentrations are almost the same, either from the plasma and the mononuclear cell concentrate. But if you compare then all these parameters, there is a comparable between group A and group B patients. So which is depicted in the diagrammatic bar diagram, where the group A, group B patients, age group, hypertension, presence or absence of the diabetes, Clelips class, and the TLR4 concentrations are the same. So if you analyze them for the primary endpoints in the group A and group B patients, a group A patients has 3 patients as the primary endpoint. One patient had an angina, one patient had an LVF, and one patient died because of the re-MI, cardiogenic shock, even on the IABP died. And whereas here, instead of the same death, one angina, and instead of an LVF, the one patient had a re-MI, but he is survived. So if you calculated the group A and the group B, definitely the group A people have the TLR positivity more, that is 11 patients when compared to the 6 patients. As expected, it takes some threshold level to be reached to come into the plasma. Then only it will be detected. That's why the detection in the group B is less. But it doesn't alter our primary endpoints, because the primary endpoints what we are expecting is at the higher levels of the TLR4 than at undetectable lower levels. So even though we detected the plasma, if they are present, that itself is a prognostic indicator for them, because they are able to detect the primary endpoints. This is the CPK values which are not different in the group A and the group B, and the primary VT, there is no difference. Anyway, we are not expecting that that is going to be then alter. If you combine then all the cases, the 17 patients showed including the group A and group B, 17 patients had the TLR4 increase and whereas 23 doesn't have it. Whereas this is in correspondence with the Nichols class, which is statistically significant, and the primary endpoints, which is statistically significant, whereas the CPK and the primary VTs, this is not a reliable one. This is the box diagram, the box plot demonstrating who has a TLR positivity has then a higher Nichols class than who are the negative. So we compared this whether is it more sensitive than the already known the risk factor, that is the cardiac enzymes and the HSCRP, whereas the TLR4 positivity, which has a statistically significance between where the events occurred versus no events occurred, whereas HSCRP and CPK, they are not able to distinguish it. Probably these are the better markers for the long-term management than in the in-hospital mortality. This is the box plot demonstrating in-hospital events are having the higher toll receptor concentration than without in-hospital event patients. So in conclusion, detection of the TLR4 is possible in the plasma, need not to be from the lymphocytes, as all the previous studies are demonstrated from the lymphocytes. TLR4 positivity correlated with the primary endpoints, which is statistically significant. When compared to the HSCRP and CPK levels, TLR4 concentration is on a better indicator of the in-hospital events. Therefore, we may use a kit to design using the plasma of the NACUTAMI patients for detection of the TLR at the bedside, and may be useful in prognostication in hospital and for a selection of the patients for the TLR inhibition therapy after the larger population study. These TLR inhibition therapies are already tried in the case of the few of the inflammatory markers like the hepatitis B and C and then bronchial asthma, which is not commercially available, but Pfizer is then working on that. Thank you.